to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 84th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for around 30 years. I'm here with my co-host Dominic Guarino.
Hello, my name is Dominic Guarino and I'm a subject matter expert in ligand binding assays and biomarkers. I've been here at KCAS Bio forever, but been in the field for a little over 30 years now.
Dominic and I are members of the growing scientific advisory team at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio's services. Today we're here with a guest, Bryan Parmentier, Research Fellow at KCAS Bio.
Good morning. Thanks for having me, John. Don.
We are thrilled to have you listening to the 84th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcasts, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources and then a focus on the topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we're thrilled to have you here and we are looking forward to a fun episode today. To kick us off for podcast number 84, Dominic is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, so as normal, we'll have our news and resources. We'll go over a few articles of our choice about what's going on in the industry in the field. And then we'll get into our main portion, which we're thrilled to have Bryan Parmentier, our guest. We'll be talking about the challenges of small molecule LCMS development and evolving approaches to ADC payloads. Okay, so we're ready to kick off the podcast with the news and resources. What's the first article we got up, John?
First thing we have is DMD gene therapy uses cells to stitch together mRNA in mice. This is something that came out of the University of Rochester. Basically, the overriding problem here is that some of the gene therapy for specific diseases is tough to approach because the gene is too large to fit inside the delivery vehicle. So this team at University of Rochester have basically looked into typical cell biology and taken advantage of that to deliver muscular dystrophy related genes to mouse cells in two pieces, which the cells, as part of the natural biological process, then stitch back together in a functional gene, and then then that leads to treatment. So where we've talked about CRISPR and other things in the past. This technique is called stitcher, and this, like I say, for the DMD, it basically they delivered functional dysferlin, functional dystrophin, muscle proteins to mice, so that those proteins are up at the normal level, and then the cells pulled those together to actually to address the underlying cause of disease.
Yeah, John. What a fascinating kind of science this is, right? So it's really new, right?
Yep.
It's treating rare diseases. So I want to take a small step back. Rare diseases are really tough to treat, right, John? You can't just give a pill. You can't just give a biologic, right? These are genetic diseases. And they're really targeting these monogenic diseases, right? And and in theory, you got to kind of treat every muscle in the body. And even now, I didn't think in our lifetime we could come this far with CRISPRs and things of that nature. But here, so you've got the underlying scientific or disease state that is let's call it tricky or challenging, right?
Yep.
And what do? They take these two AAV viral vectors, and we've talked extensively about them on this podcast here, and then they slam them together to introduce these two genes, and somehow magically they inject them into this mice, and the mice's ribosomes pick it up, and somehow these animals are producing these proteins in their active form, functional form. I think that's important to point out, and that's a big bioanalytical challenge too, right, Bryan, John? And so then magically, I still don't know if we've truly grasped just how this works and what some of the side effects are.
Well,
But this looks promising, right? This
yeah,
stuff works. It
yeah.
is awesome.
So the lead senior author of the paper, Douglas Anderson, he basically says they don't know the exact details of the repair pathway. So it's basically like you say, it's two vectors. Each genetic sequence contains part of the final gene attached to its ribozyme sequence. Once delivered to a muscle cell, the gene is transcribed into mRNA, and the ribozyme activates, cutting itself off at the strand, leaving behind the chemical footprint the cell recognizes. With both halves in the same cell, the repair machinery springs into gear and stitches the two strands together into one mRNA, which is then translated into a functional version of the protein. They have identified that RTCB ligase, which is a RNA repair enzyme, is involved in the process, but like they say, they're still trying to figure out the process. It's just, to me, they said, like a huge step forward. We know that there's a lot of focus on the Duchenne muscular dystrophy, and a number of there's a couple of approved
Hmm.
medications out there, but a lot have stumbled in these final clinical trials where it comes to motor function and things like that. They just haven't delivered. So maybe this alternative approach is a way to address it. So
Yeah.
yeah,
It's really cool. I mean, they mentioned Sarepta, and they have a gene therapy, but I think they're overcoming not only some of the challenges with treating rare diseases, but that had a dystrophin gene that they called a microdystrophin. It wasn't kind of as big because of the size of dystrophin, right? That's another
sure.
challenge. It's
Yeah, and that's the fundamental thing here. It's using this as a mechanism, not just to address Duchenne, but
yeah,
also it's this you know delivering things in separate parts, stitching them together, and that's what's the potential for this in other conditions. So
yeah.
it's a really nice way to start things off.
Yeah, I mean, I guess we should maybe give a shout out to this Bridge Pharmaceuticals, which is kind of funny, right?
Yeah, yeah,
Based
they're based in China. Yes, yes,
yeah,
yes.
but I Bridge. That's kind of cool. But no, and then Rochester, up in New York, great school, John. Right,
Yep.
I can go back to my graduate days. I partnered with someone up there with some HPV work for cervical cancer. So, but cold, I'll say that about Rochester. Great school though, but great stuff, John. I think it's amazing, and you can see how they talk about monogenetic diseases, but there are many that are multiple sorts of genetic diseases, and this could sky's the limit. Maybe they could maybe it could be 4 or 5 different viral vectors, right? They don't really talk
Sure.
about that
Yeah.
here, but
Yeah.
it has huge upside, and just it's early stages, but that's what we talk about here. And I think you kind of said it real nice early on when you said, "Hey, this is like the next kind of version of like CRISPR," because
Mhm.
CRISPRs are first generation. Well, maybe they're like the ones that are going into people are now third or fourth generations, right? CRISPRs are actually 20 years old, but they've just now made it into the clinic. But this seems to be, as you said, a pretty significant step up in terms of
Sure. Yeah.
targeted gene therapies. But I'm sure we could talk more about this. But it's a really cool kind of academic. But these are the things that in 4 or 5 years we'll be hearing about.
Yeah. Because
Five, right? These are the
like you say, this is done in combination with Cambridge Pharmaceuticals, so that they're obviously looking at a commercial path for this. So it'll be something to watch out for. Let's move on.
yeah, cool one here too. Fun
So
thing, our
the
friend.
next one, it's this. I wanted to come at a slightly different angle. Obviously, we have a lot of chronic diseases around the globe. One of which is tuberculosis, which is tied to one and a quarter million deaths in 2023, and is likely retaking the position of the world's deadliest pathogen back from COVID-19, according to the World Health Organization. Current treatments involve antibiotic regimens that take 4, 6, or 9 months. I was vaccinated for TB back when I was in high school. It was the BCG, which was the vaccine at the time.
Is that the arm, John? So you got a
Yeah,
little scar
it
on
was,
your arm? Yeah.
you basically you were the process was they did a punch on your forearm to see if you needed the shot or not, and it was like a series of 6 punches or like jabs, and if that swelled up, then you didn't have the jab. If it didn't, you had the jab. But then the BCG was one. It took a long time for some of us for that injection site to heal. So it was months before mine finally healed up, and you'd be doing any sort of physical activity. If someone hit you, it was a really painful injection site, and then it would just, you could easily take the top off and you'd be bleeding everywhere. So yeah,
we can talk about, John, that's like its own thing, because you just described like a Whalen-Fleur reaction, which I don't even think they teach that anymore, really. I don't know if they do a lot of what you just said, but let's talk about this article. It's fascinating.
So that's an aside. Anyway, so this is actually, so the headline was compounds from bog fungi kill tuberculosis bacteria in the lab. So again, this is really early stage, and they're sort of saying that actually the compounds they identify aren't applicable, but at least they could point to a treatment approach in the future. And so basically, it's researchers at National Institute of Health's National Institute of Allergy and Infectious Diseases turned to the natural enemies of the pathogen that cause tuberculosis, fungi living in peat bogs. 3 compounds isolated from tuberculosis fungal foes killed culture cells of the bacterium that causes the disease, and the bacterium is Mycobacterium tuberculosis in lab tests. So this was published early December. They actually screened somewhere like 1,500 fungi species collected from sphagnum peat bogs in West Virginia and Maine, alongside the tuberculosis pathogen. And so fungi in these moss-filled bogs, alongside Mycobacterium tuberculosis, they're in the same genus, and they compete with Mycobacterium tuberculosis for the same resources, and that's how the fungi kill it, because if they're superior, then they knock it out. So what they found of those 1,500 fungi, 5 actually were active against Mycobacterium tuberculosis, or MTB. I'm going to call it that. It makes it much easier. And the 3 compounds that they found effective were patulin, citrinin, and nidulanin A. Were the toxins to the bacterium. So like I said, they said the authors note that these toxins likely wouldn't make good drugs themselves, but their activity in the thiol pathway suggests potential targets for new TB drugs.
Yeah, it's a fascinating read, John, right? So tuberculosis in and of itself is like a devastating disease, one that is incredibly invasive. Part of it is similar to, I kind of think of it, it has like a lot of life cycles within your body, and it makes it really tricky to kind of get there. It's rugged, it's hardy, it isn't always deadly, right? It's brutal, and it's been killing people for decades, centuries.
Sure. Yeah. Well, I mean, I was just thinking about it as you were talking. You think of it as in terms of literature from like the 19th century and early 20th century. The number of people that died in TB is part of the story. It's because it was a constant feature of life. So yeah, it is a big deal.
And then it's like shocking. I mean, we just forget how great it is here in the States.
Yeah,
right? I
we
mean,
do. Yeah,
or even like the UK too. But like tuberculosis kills, it said 1.25 million people in 23. I don't think any of them in the U.S. I really don't. I don't. I think we're still blessed that there isn't that, people die, and sometimes I've recently read about like an outbreak here. Someone brought it in, but like we're just fortunate. But it makes me wonder. But back to the therapy. It's so cool. So is peat part of like whiskey and scotch and things like that? Am I making that up, John?
I'm not sure what the process is. There are.
So I bring that up because it's like, just think about how great science is, right? All the tools to treat us, all the medicines, even though most of it's chemistry based, they're all coming from things that somebody naturally kind of produced, and then we make a recombinant version of it, right? Or a synthetic version of it. But it just brings it back to like all the tools are there. We just got to figure out how to unlock it. And that's why your diet, right? That impacts everything. I mean, diet certainly has an impact on your overall health because the building blocks and so forth. But these types of stories are great, John. This is classic I think of it as like basic research, the true research side. These are people who are scanning what they say 1,500 different fungal species. They identified 5, right?
Yep.
Had toxic effects, and then they go straight into this thiol reactive. That's what these things are, right? So it's awesome, man. This type of stuff I don't know if you're not if you're a true scientist, I'm just getting all energized because it's like, I don't know. I haven't done this type of like kind of basic research in years, but this tools us. This is where it's at. These are the things that are going to help us with antibiotics, right? This leads right down that pathway too. And then just I'll end with, if anybody knows the history of like biotics and medicines, a lot of it came from burning oil and blubber, right? Whale blubber. And they were collecting things bare. That's how bears found it. They're collecting things in the corner with swabs, and they're trying to identify what's in the soot and everything. And they found out it's killing cancers, and half your medicines are coming from stuff where they were just like literally things that were coming out of byproducts of stuff that we were using. And this is another example of it. It's awesome. And we started on like the rainforest because everything's in there as well, right? All this medicine man type scenarios. But I don't know, it makes me warm and fuzzy to think that there's somebody out here working on these things. Just like we're not going to die of antibacterial resistant bacteria anymore because there's a wave of those coming. This falls right in line with this. It's amazing. Tuberculosis. The reason why tuberculosis isn't really probably cured is it gets back into, it's not a moneymaker, John. But we'll move off of that, right? So I don't know if you want to end any thoughts, but this type of stuff is great. If you're a scientist, I hope if you're a young scientist, man, there's nothing better than just working on these. You think, oh wow, who would ever use this stuff? No, this is the backbone of it. Yeah, this is it.
I think that's it. It's that recognition of I'm trying to think, recognition of a natural relationship, and then how that natural relationship could possibly be translated to help us. But then, like you say, the amount of legwork that has to go into it to get from the recognition of these fungi that coexist with MTB, but could give us a pathway forward, and then building on that. Yes, you're right. The basic research stuff is fascinating, and that's why I love finding these stories about how natural products can help us. And there's lots of examples. I mean, we got Taxol from the yew tree. Yeah. We know that's one that helps us. We got Bayetta, the Gila monsters. Saliva that then led to the GLP-1 analogs, things like that. It's so fascinating.
John, fruit flies and zebra fish, all those things have been researched. Those are all, and that's another thing about TB. I'm like, what do I know about TB? A lot of the immunology, my doctorate in immunology, a lot of the models that we learned about were TB driven. Sure. We learned so much about the immune system because it took advantage. Tuberculosis has certain things that it releases that go from acute to chronic. And when you go from the acute cytokine profile and cellular immunity profile into a chronic version, the bug survives. Yeah. Right? Because you're going from that pro-inflammatory, really, trying to kill everything around it to, hey, this stuff's okay. It's supposed to be here. It's normal. That's quote unquote, like the T helper 2 response, right? I don't know if you know what I'm talking about, but you study T helper 2 because tuberculosis helped produce tons of IL-5 and other things to assuage the immune system away from acute into chronic, just like a tumor does. Sure. So like these things are all kind of closely related, and this is. Sure. I don't know. I guess I'm really happy I did a couple stints in really basic research labs when people were like, what are you doing? And I'm like, nah, this is how you learn stuff. Yeah. Dropping genes from stuff and all sorts of unbelievably weird stuff. And let's maybe move on, though, John. I want to be cognizant of our guest. And that was a cool article. And we got
a little news item that we'd cover in like a minute, but hey ho.
Yeah, no, it was good. That's a good one.
That's it. And then we're back on the GLP-1 analogs. But this is still to me, this is fascinating because we've got definite indications for the GLP-1 analogs. We're seeing label expansion into new conditions, but because of in this case is a retrospective analysis shows potential for GLP-1s to treat blood clots, and this is looking at, again, this is basic research. It's digging through data and seeing additional effects from data that already exists and really interrogating the data that you already have. So this was basically a retrospective study suggesting drug classes' potential to treat venous thromboembolisms or blood clots in the veins for individuals with type 2 diabetes. So this group, I'm trying to see where it was. I'll find where the group are in a minute.
Yeah,
Basically, they looked at electronic health records from hundreds of thousands of diabetes patients and compared those who took GLP-1s in combination with DPP-4 inhibitors compared to those who took DPP-4 inhibitors alone. And what they found was that those on GLP-1 drugs had a 20% lower risk of developing clots after 1 year. That group also had a 22% lower rate of pulmonary embolisms, that are clots blocking arteries in the lungs, and an 18% lower rate of deep vein thrombosis. So basically, the DPP-4 were chosen as a control group because they used a lot in diabetes, but had no known effect on venous thromboembolisms. Nice thing about this is they weren't really trying to drill down on a particular GLP-1 drug. There were patients who were on all the approved drugs to date. So we talk about semaglutide routinely, we talk about tirzepatide, but also include things like Byetta, the exenatide that we cited in the previous discussion. And also there were people who were on metformin and insulin for comparison. And so it's just, like I say, we're still figuring out what GLP-1s can do. It seems like they're miracle drugs, and there's obviously things that are probably going to fall by the wayside. But it is very interesting, the interaction of these drugs with body pathways and how they can be very beneficial just beyond what we found for them so far.
Yeah, no, John, it's these retrospective studies, I believe, on these GLP-1s that are becoming somewhat ubiquitous, we're going to keep learning more and more about. But this seems like there's 900,000 cases a year of venous thromboembolisms, and this could be something that could prevent upwards of a quarter or a third of them, right? That's kind of what the data says. It's still hard to grasp because I don't know what this DTP4 inhibitor was. It's an interesting.
I'm trying to think what the drugs are. They're small molecule drugs.
Yeah, it's
a whole class of drugs, and I can't think what the name of any of them are offhand.
I think the most impressive thing, all study designs are important, right? That's stating the obvious, but the fact that they put a control group and then they just took every GLP-1 on the market,
sure,
it's important there, and as well as any GLP-1s. Which were approved in the mid-2000s. I don't think I know what a GLP-1s is, John. I'm going to show some ignorance there.
Me neither.
I can't quite tell what that is. But the fact that you get all these things and the data is correlated is pretty strong, right? That's amazing. I think I wonder if I don't know if, I didn't see it in the article. I wonder if they stratify it by the drug type to see if there's, maybe one group is 35% and one is 15% or something like that. They didn't cut it that way. But this is great, John. I think this is the same thing as like Viagra and Cialis are helping with heart attacks and strokes. The numbers there are unbelievably strong there. So I guess, John, in another 10, 15 years, I'll just be popping a pill for helping me prevent a stroke, and I'll be jabbing myself in the side once a week, make sure I don't get any blood clots and my heart doesn't go out, right? I guess
there you go. Yeah.
Body biochemistry, I'm embracing it, John. I got no problem with taking all these things should it be needed, but I don't know. I think it's real promising, and knock on wood, the side effects for GLP-1s have been minimal. That's kind of cool.
Yep.
At least there was some, some articles published out there that were kind of trying to indicate, but I haven't seen anything concrete there other than, it just doesn't make food taste good for certain people, and that is no way to live. I guess that would be hard. I love food.
Discussion to the end.
John, I don't really have anything to add. It's just the GLP-1s are here; they're not going anywhere. I bet you the next decade these will be blockbuster drugs. But just like we just learned about the next wave of CRISPR, I'm sure there's something somebody's cooking up that'll.
Well, I think the next generation for the weight loss piece in these GLP-1s is the oral forms to make it easier for people to dose. Anyway,
I wonder if the GLP-1s are like the fen products from years ago. Remember, those were weight loss drugs too.
I really don't know.
Yeah.
Anyway, if you're curious about DPP-4 inhibitors, they basically The chemical names are things like sitagliptin, saxagliptin, linagliptin, and alogliptin, and may know brand names from
Yeah, those sound familiar.
Januvia, Zituvio, Onglyza, Tragenta, and Nicina. So I don't know if you've seen ads or whatever.
Yeah, those are diabetic drugs. Yep.
Yep,
Okay.
so they work by blocking the action of the DPP4 enzyme, which helps inactivate GLP-1 and glucose-dependent insulinotropic peptide GIP. This allows the body to increase insulin levels after meals, which moves sugar from the blood into tissues and helps keep blood sugar levels stable. So yeah, they're well-established
John,
diabetes drugs.
you just mentioned GIP1 there. I wonder how much we really know about this pathway, right? But
Well, I think that's what's going on.
Yeah.
When you say next steps, I think it's learning. I think it's still in investigation mode on this.
It's the negative feedback loop of it is fascinating stuff. But the circadian rhythm, right? That's all kind of tied into this. I wonder how much we really know, but it's really cool stuff, and certainly plays a big role in how you feel after eating or throughout the day. Your pituitary is a big part of it, as well as stuff your pancreas makes, obviously, for food. For food.
Let's move on.
Yeah. I'm babbling a little bit.
Yes, it's fine.
Harmless skin bacteria. So
I'm
Bryan can jump in and contribute. Yeah, tell us about this bacteria, which is really another cool one, John.
Yeah, exactly. It's between podcasts. I harvest news and try and figure out what is actually going to make something interesting to talk about that isn't a merger or a drug failing in development or things like that.
Oh, there were plenty of those this week too.
Oh yeah, and this is amazing because like I was sitting this month thinking, oh, we're going to be sitting twiddling our thumbs. There's nothing to talk about, and all of a sudden it was like this flurry of things that, oh god, that's fascinating.
I did think of you. I'm like, oh, these are pretty esoteric or specialized. Yeah.
And so here, the headline in this is harmless skin bacteria engineered to be living topical vaccine. The more I read about this is incredible, and this is published in Nature on December 11th. And the lead author is a Michael Fishback as a bioengineer at Stanford. And what they did, researchers engineered a common skin bacteria to express a tetanus toxin on the surface on the skin of mice. When mice were colonized by these bacteria, they developed immunity to tetanus that prevented them from dying or showing symptoms when later exposed to the toxin. What they did was they took Staphylococcus epidermis, which is a skin bacteria. It's harmless and resides on every hair follicle of virtually every person on the planet. They found that these microbes are abundant and don't really do it. That doesn't really disturb anything, but they do induce a T-cell immune response in mice and primates. Fishback and colleagues determined that B cells too are involved in the immune response to Staphylococcus epidermis. Mice swabbed on the head with the bacteria generated antibodies against accumulation-associated protein, a large protein that sits on the surface of the bacteria. So they swapped fragments of this as accumulation-associated protein AAP with antigens from tetanus and diphtheria toxins, and then letting the bacteria colonize mice skin. And the rodents' B cells generate antibodies that then target the pathogens. And so they found with the tetanus, even at 6 times the lethal dose, the mice were immunized and fine. I find this like, holy God.
Yeah.
This is amazing.
It is. It's like a scene out of Star Trek, or even, I don't know if you remember the Transformers where one of them has like a robot literally building his face back up. It's it'll come to me who the evil Autobot was there, or sorry, I'm confused.
It's after my time.
Yeah, it was. I'm confusing but it's it's vivid in my mind in terms of, John, this is really cool stuff. So I'm very much into trying to preserve my skin because of the sun, right? I'm bald. I got to be careful of that. I've been trying all sorts of creams and stuff. And the thought of being able to put something on, a lotion on, that can engineer like a humoral immune response is just awesome. I just envision a day where you're putting SPF 30 on to moisten, give you a little moisture on your skin. Oh, and by the way, it's a cocktail of COVID and influenza A and everything else. You get a whole bunch of antibodies being produced at the same time. Now that's a big leap, but John, again, this gets back into our grandchildren someday might be shopping for this type of stuff at the maybe not the mall. I guess whatever version of things there are back in time, but this is topical stuff is so powerful. There's been a lot of barriers with it that have overcome and just in terms of like the last 40 years, 50 years, understanding Langerhans cells, which are like just understanding the dermis and the epidermis. It's kind of its own, like it's the largest organ, right? And it has its own sort of immunological kind of aspects to it. And all that research that I think people have been doing has found its way into some therapeutics. I still can't quite figure out the mechanism of how this is happening, but that's not I don't think it's really that relevant right now. But I just, John, this is incredible stuff. At the thought of like, oh, hey, just like I said, oh, you don't need to get a vaccine anymore. Just these topical things.
Yeah, well, that's. I was just thinking about this. I mean, given how accident prone I am and the number of accidents I have on a weekly basis, if I could just, the risk of tetanus, because I've impaled myself on a rusty nail again, I could just put a cream on to help address that. That's fantastic.
Think about it. You could diphtheria, any kind of. Well, we don't really have these things, but it could help like global health at an epic level.
Yes,
right?
exactly. That's exactly. I mean, that's where you see the potential. It's less for idiots like me who keep impaling themselves on things.
Think about it,
as opposed to a much wider population.
We forget how easy it is to get a jab or get anything, tube of blood drawn, and a lot of places that's even a pharmaceutical. Man, the idea of just what you said. Oh, hey, impaled my foot with something dirty. Okay, don't need to go to the hospital anymore. Just bandages up, rub this on, we're good to go, right? That type of mindset would be fantastic. Or even just, I don't know, the sky's the limit. My mind's racing around, John, but we're going to close. Why don't we move off of this? But
I just want to say, because we have mentioned who the group were, but they actually do have a plan as to where they want to go with this. So they want to see if they can replicate what they've done in mice and primates, and then they're talking human trials in 2 to 3 years from here. So it would be very interesting to see where this goes to.
Yeah, they got to do some more engineering to get it.
Oh, absolutely, yeah.
But it's Stanford, right? Where else? Stanford's fantastic. It's great.
Sure, exactly. So I just thought, again, it's always nice to touch on this early research stuff and trying to figure out the potential of it and how it can change our lives. And some of these things will definitely have legs. But yes, let's move on to the main topic.
We got a special guest. We'll jump right in, right, John, if it's okay?
Sure, go ahead. Feel free.
Yeah, we got our good colleague of both John and I, Bryan Parmentier. Before we get started, maybe Bryan, just give a quick hello, say hello. And
at least say what the topic is.
Yeah, sorry, my bad. Go ahead and tell us the topic.
We brought him in to talk about something of his choosing. No, we actually did direct this. So we want to talk about challenges of small molecule LCMS development, and we're going to touch towards the end of it about where we're going with ADC development validation, the free payloads, which is the small molecule part of this. So we presented some of Bryan's data at the recent EBF meeting, and I think this is because it actually affects how the group work. Then that's something we want to talk more about. But that's all within the context of small molecule method development as a whole. So now we can turn it over to Bryan to introduce himself. I don't want to steal all his thunder here.
Good morning. Thanks for having me. I'm looking forward to the talk.
Yeah, great, Bryan. Before we dive in, I think it's important to talk a little bit about, we if you've been listening to this podcast, small molecules are not dying. They're still, I still think, John, I haven't seen the numbers this year, but is it 60-40 split roughly still small molecule approvals? Still dominating. Go ahead.
That was something I meant to check before. Yeah, because I was going to hype up small molecules.
I still feel like it's. They're always.
they always have they're always a big feature in terms of approvals per year, and yeah, they'll be up there. I think we talk so much about large molecules because they're the interesting chemical entities. There's more challenges to them in terms of bioanalysis, etc., etc. But you cannot ignore the small molecule piece because it is a vital part. So yeah. Anyway,
and that's a good point, John. Right? You bring up two good points, and then we'll let Bryan dive in here. The evolution into the ADC space is definitely where most people think small molecule engineering and advancement's coming from, but that would be a false statement. There's still plenty of new compound entities, and then I'll add. The bioanalytical challenges of small molecules have not gone away. And that's really where I think Bryan's going to kind of dig in, because people often just think, well, it's just the payloads on there, and it's the same as if I was doing it over here as just a standalone payload. Well, no, we know that's not true. So let's dig right in. And Bryan, I don't know, John, how we want to take this.
Well, I think let's give Bryan a chance to talk for a start. I think we're a little bit fired up.
Go ahead, Bryan? Why don't you tell us a little bit about what you're working on?
Tell us, Bryan. As an introduction, tell us what your current role is, what your background is, and just what you do on a daily basis, and we can then start going from there.
Hey, good morning. So my current role at KCAS Bio is I serve as a research fellow and also a principal investigator within the small molecule LC-MS method development department, and I oversee a team of MD scientists. We cover everything from non-GLP discovery all the way up to development for GLP validation assays. Did you need anything more than that, John? Or?
Yeah, where were you before? I mean, I know, so a bit of background, Bryan and I started on exactly the same day at KCAS. So when we're in calls and one of us says we've been here 5 years, the other one goes, yeah, I've been here 5 years too, because it was exactly the same timing. But yeah, I think, it's always good to, when I interact with Bryan on calls, my background is more in the validation end of things, and so it's always useful to have a scientist who's got really thorough method development experience, because that's when you're talking to customers, that's what you want to get across. It's the depth of knowledge. And so ultimately, although we've both been 5 years at KCAS, what is actually more valuable is the amount of method development experience prior to that. We're both getting up there in terms of age, but it's what we've done beforehand that is the value, and how we help customers out.
Okay, yeah. So I've been working at KCAS Bio for 5 years, as John said. Previous to that, I was at Charles River for approximately 24 years. Came out of school and got a job as a medical technologist, so worked in clinical pathology for about 5 or 6 years, in clinical chemistry, also hematology and urinalysis and parasitology and coagulation, and a hand of other things. I was always volunteering for out-of-the-box assays when they came along. Through a friend, I wanted to get into forensics eventually, maybe get into the police department back then when I was in my young 20s. I learned that I needed to have some mass spec and HPLC experience also, so ended up segueing into the LC-MS department at Charles River, and I went into the GLP department. Went mostly into the method development and validation area. I kind of like that more than sample analysis. Worked with some great scientists there on some very hard projects pretty much right away in my career. Was in GLP a number of years, and then had the opportunity to get into discovery work. Worked in discovery also for a number of years, both small molecule and some large protein work, all by mass spec, like in the early days of Skyline. Then went back into GLP after that as a very experienced, I would say, mass spectrometrist by then, and back into GLP, but then I was more overseeing GLP staff, but still working as a principal investigator, working with the clients. And then the opportunity came along for me to take all my method development skills and lead a team at KCAS Bio, where I am now. And that's my experience.
No, and we're so fortunate to have you, Bryan. I could ask you a whole bunch of questions about things, but we don't. For time's sake, But you've had an illustrious career. We're so blessed to have you here. You do solve a ton of problems for us, and you do it with a smile, Bryan. I think that's important, and you're a good leader for our organization. But so we're gonna jump right into our agenda here, Bryan, and we're gonna, we can take it any way we want. But why don't you paint a picture of just what's a typical small molecule method development, right? Like okay, you got your materials, you got your deuterated internal standard. I don't even know what I just said, by the way, Bryan. I don't know if that really is, but you got your internal standard. So how are we how do you tackle a small molecule? And I know maybe without giving you, this class of drug and this class of drug, but if I'm a small molecule scientist out there, what am I looking for in terms of developing a robust method? Maybe that's the way to ask the question.
Sure. So if we'll say we're going to develop a robust method, say for validation. So we're going to start off, as you said, with the analyte and hopefully a labeled internal standard. If not, we take whatever steps we have to get it developed. We develop chromatography first, and develop the optimized sensitivity, linearity, carryover. We look at accuracy and precision. We look at multi-lot selectivity, multi-lot matrix effect. Now we also look at benchtop stability and freeze-thaw stability to cover how the samples are handled in the lab. We look at analyte and internal standard recovery. We looked at dilution integrity in case we have to repeat any samples that are above the quantitation limit. We look at re-injection stability in case we have to reanalyze a plate or specific samples in an analysis run. And we also look at whole blood stability, and we mimic the collection of the samples to make sure that they're handled within a time frame that the drug is going to be stable during the collection.
So to go back to early development, do you do much in terms of standard solutions, or you try and get into matrix as quickly as possible?
We also do look at, primary stock stability in solution, and then spiking solution stability, and then once in a while in development we'll look at the internal standard stability if something's suspected.
And so when I'm talking to people at development or about LCMS in general, I mean, you basically got three points where you can optimize a method: extraction, chromatography, and the mass spec piece. In terms of extraction, what's your drivers for, if you're given an analyte on paper and this is the structure and this is the detection limit, what drives your direction for where you go with extraction? Is that upfront piece, and then, because obviously the chromatography is one piece, and then the mass spec is going to behave, the compound is going to behave a certain way regardless. So how do you approach that?
We mostly focus on high throughput whenever we can, right? So we want to have the shortest runtime and the easiest extraction with the quickest turnaround. So we're usually centered on protein precipitation if we can. And if we see that the drug class, say, has a log P where the drug is more hydrophilic, and maybe it's not going to extract by protein precipitation, or there's some known interferences in literature, say from lipemia, or we're doing a study where, say, it was an IV and we may have a vehicle that could interfere, then we take other steps ahead to come up with a different kind of extraction. 90% of the time we want to do something, the easiest that we can.
Sure.
We of course have to make it through those validation assessments. We usually touch on some of them very early on before we actually get into the formal testing. So,
Yep. So it's interesting, John, because you started asking some poignant questions, and me as a novice over here is like, what did he mean by chromatography, and what is carryover? So it's funny how different we are in things.
Sure, yeah.
And we're sort of getting, but I do think it's important for time's sake, and we're moving towards more agenda topics. Bryan, you started to scratch the surface at some of the biggest challenges for mass spec, and John, I don't know if you had more around development and so forth, because my questions were really like, and I think I can save them, because as you talk about the challenges, Bryan, keep in mind some of our audience doesn't truly understand when you talk about chromatography challenges. Maybe I'll just ask that question. Give us a little bit about what you meant by chromatography, and what are some of the challenges? How do you troubleshoot that? Does that sound like a fair question?
Yep, absolutely.
And Bryan, I'll remind you to yell at us. I'm a little deaf.
Chromatography challenges usually arise when you have a solubility issue with the mobile phases that you're using, or the drug in the injectable solution, where it's not optimal and it's not reproducible on the column, or not retentive enough, and then we step forward to optimize those accordingly, so that we have a K prime of 2 or greater.
And just to step in, the K prime is basically a measure of the amount of retention on the column. And it's basically the K prime is related to the retention, or the amount of time your analyte stays on the column compared to the void volume, where the void volume would be something that's not retained at all. So for us as chromatographers, we want to be in a certain K prime window, because if it's too low, then you're not retaining things, and you're going to be more subject to background interference. If your K prime is too high, then you're basically retaining the compound too long, which doesn't go into the nice realm of rapid turnaround, because you can't get your compound back off the column, and so your cycle time is way too long to have a efficient throughput of samples.
Interesting. Wow.
Thanks, John.
Yeah.
So that's one side to some of the optimization, so that could actually be solved in the solution itself, and then the other challenge is working with the extracted matrix sample. So the baseline of the extracted sample could have different species or interferences in the baseline that interfere with the drug. Therefore, we have to adjust the gradient in our approach to get around those interferences.
And Bryan, does that translate itself into noise is that what you would call that, right? Or
baseline noise?
Yeah. It'd be noise, or it could be an interfering peak as well. And
whoa, hang on. What's the difference? Sorry, what would be the difference between is an interfering peak a form of noise?
No, noise is more background. An interfering peak could be something that actually elutes, i.e. comes off the column close to your analyte, so it actually distorts your peak shape. It was something else I was going to say, and I'm not.
Sorry, I think that's I get it now. So it's not just an interfering peak means you got to troubleshoot one way. Noise is like, I can troubleshoot this way, right? Is that kind of okay? Got it. I hope
one more thing. Because chromatography, the purpose of our chromatographic step, which is a separation step, is to try and get, because like Bryan says, the deal is you've got a complex matrix. You're trying to get your analyte away from all the other garbage that comes out in your extraction, because you're not going to just extract for your analyte. You're always going to get co-extractants. And so the chromatography is trying to ensure that you've separated your analyte from all the other garbage.
Yeah, yes. Bryan, add as you see fit, but we'll fire another question at you here in a minute. Please share your thoughts.
Just add one more thing, is as far as noise, we often have to isolate if that baseline is coming from the instrument or coming from the extracted sample itself.
So I want to press you, dig in on that. So you said it can come from the instrument, i.e. carryover? Is that what that would be? Okay.
No,
We'll get to that
Oh
separately.
my God, my head's spinning. I'm just gonna put myself back on mute. Let's continue with any. So we were talking about challenges. We maybe, do you have other challenges you want to share, or other things, Brian or John, that might be important or good for the audience to know around, just like, hey, I'm running LCMS. I feel your pain.
I could describe some of the biggest challenges. It's usually matrix effect. So, the phospholipids in the extract, or hemolysis, or once in a while we'll have some endogenous species that are in the sample that will interfere. And also we could have drugs in the sample. They could be co-dose drugs, or we've had instances where, say it was a prodrug study, there was interference from that, or what we see these days is ADC interference, or a conjugate will interfere, the larger part of the molecule.
And other issues that we'll see, to answer your question, Dominic. It's one reason why it's important for us to know structure up front is looking out for the potential for unstable compounds, so we may have to stabilize them in process, and that's one thing. A carryover is another big issue to deal with, and there was one other which has completely escaped me.
That's okay. I had a couple questions for Brian, because he said some cool stuff.
Yeah.
I'm loving this, Brian. We could be here for a long time. We got like 5 more agenda items, and I got 2 more questions related to some of what you said. But maybe we this could be a long one, or we'll have you on again. But yeah, so one is, you mentioned phospholipids. To me, phospholipids can be they're ubiquitous. And then the other thing is, like, we still don't have a good grasp on, like, what's the level of phospholipids in my cell lysate or my matrix versus yours, right? I mean, these things and they fall off. There's still, up to me, phospholipids, phosphoproteins. These are like bioanalytical headaches. I basically tell our clients, like, sure, we can take a look at it, but take the 20 grand and light it on fire approach. I don't want you to be mad if we can't solve this problem for you. 4 other labs have tried it. Sure, we got expertise and we're great, but hey, that's not shooting fish in a barrel, and I want to set the expectation the right way. So phospholipids, why? Is it just the charge gets in the way? What is it about phospholipids? And is that like a lipemic sample that'll have a lot of phospholipids?
Yes, there'll be greater content of lipids in a lipemic sample, but as we were talking about earlier, when we're designing the extraction, usually we'll see issues with phospholipids when we're looking at multi-lots. We'll see that some lots don't pass criteria, and that points to that we need to use a better extraction. So instead of protein precipitation, we'll need to clean up the sample more with like solid phase extraction or liquid extraction, or optimize the protein precipitation extraction more to get rid of that.
Yeah. This isn't a problem in the the large molecule side. That's why it's interesting. And maybe you had something to add, John. But before you do that, it's like, so if I look, it's hard to prevent a lipemic sample. So if there's a high incidence, you may have to proactively adjust. You're not going to go to protein extraction, right, or whatever the standard is. You'll kind of proactively tackle it. For example, if I said, hey, we know there's going to be a lot of lipemic samples, right? There's disease states that you're trying to. But the alternative, Brian, if you're in method development and you look at lipemic from your standard and it's a problem, you then have to adjust. Is that a
yes?
Okay, got it. Thanks. Sorry, that's helping me. It's probably helping our BD team. I don't know if it's helping our audience, but a lot of that's really fascinating. I don't know why it's clicking for me better now, but and it is because the charge, right? Those the higher, not necessarily.
No, It's basically it can be they've co-extracted, and you may have them co-eluting with your analyte. You might not you won't see them, but because they're co-eluting, they might be suppressing your analyte. So if you've got variable concentrations of phospholipids between, this is why the multi-lot experiment is so important. If you've got variable lots from different donors with different levels of the phospholipid, they may suppress or change the signal of your analyte such that you're not reliably measuring that analyte in samples.
And this is where LCMS is from Venus and LBA is from Mars. That's not called selectivity in your world, or is it?
That would be part of the selectivity experiment. Yeah.
Okay.
Yeah.
Okay. All right. Great. I'm sorry, guys. I did have one, but it's about the ADC interference. That's all the rage, right? That is where.
I think we'll touch on that at the end, because it is really important.
I don't want to derail this too much.
No, absolutely.
Very educational for me, because, like, John, you're starting to ask him questions, and I'm like, man, I don't even know what he just said.
And I think that's why it's useful, because we again, I think to some extent we take small molecule LCMS for granted, but it's not. And I heard people talking about it as a commodity, and it's really not. There's a lot goes into a small molecule development.
John, when you guys were talking, I thought about the amount of chemistry you two know is pretty high. Right? You need to know chemistry to troubleshoot that extraction process, right?
Sure. And the chromatography, and
all of it. The mass spec itself is more physics, right? But
and also when you talk about interference, and matrix effect, we're not seeing the interference. We just know it's having an effect on our analyte. So there's a lot that we're having to watch out for, because the mass spec is a very selective detector. So when it comes to the actual measurement of the signal from our analyte, all we may see is our analyte, but there's other things in the background that we can't see or we're not looking for that may actually be impacting our ability to quantitate that analyte consistently for a robust assay, because obviously we want that assay to be robust and reliable, so we're giving good data to our clients, because it leads to drug approvals down the line. So I
think in my mind, right, correct me if I'm wrong, but there is definitely some of that. It's just how much can be tolerated before it affects the sensitivity, right? Because there's got to be some, and I think to me, it's like you guys are. I used to laugh, like, kind of jokingly say, you guys are making this stuff up, right? I used to be like, oh my God, look at these pretty little chromographs, and you're getting all this fancy information, and I would think, oh, whatever. But I think now it's clicking more around, hey, it's no different than the way I'm extrapolating off of a curve from an MSD assay. There's still that sort of leap of faith around, hey, these are relative. There is no, when I say relative, I mean that actual signal is a jumping point, and then there's up and down from there. But maybe we move on, right? Because sensitivity is all based on the signal, how clean a message it might be. But sometimes the cleaner the method does not make a better method, and that's a whole nother topic, at least for LBA. I don't know if that made sense, but let's move on, gentlemen. Let's move on to, how about the evolving role of small molecules in LCMS?
I threw this out because basically where I'm coming from, and I'll let Bryan talk about this. We talk about small molecule LCMS, and we're not. We think of small molecules between. I'm trying to. They're sort of molecular weight down between 200 and 500, but drug design is changing. And so the small molecule team doing intact molecules via LCMS, the type of molecules they're dealing with is also changing. So that's where we get moving into peptide therapeutics, oligonucleotides, the GLP-1s we touched on earlier. Also, the same team is also working on biomarkers, which goes into the completely opposite direction. It can be molecules that are down around 100 molecular weight that come with their own issues. And I'll throw that out to Bryan, and sort of what's the fresh and the new that you've been seeing beyond your traditional small molecules.
Yeah, so we've been seeing more and more requests for endogenous small molecules, which have their own challenges using surrogate matrices, and we have to demonstrate parallelism. have to have special handling of the real matrix that contains the endogenous species, because that has its own stability. And if you imagine, it becomes almost a double validation. You have two kinds of stabilities, both surrogate and real matrix. And then also the oligos are becoming more and more popular now. We have a lot more projects for those, and that has a more in-depth extraction, usually solid phase nowadays, with more steps and more attention to details of reagents and handling, and also the analysis. The analysis, we have to have the instrument conditioned ahead of time with the drug and the ion pairing reagent, to make sure the instrument is at an equilibrium to be able to handle analytically our run. So we're on to a bunch of new troubleshooting challenges, and we've addressed many of them recently, and have come up with great solutions, and it's getting better and better as we go on. We also have worked a lot with ADCs lately, and that brings its own challenges that we'll talk about later.
And so for oligos, I mean, you're also, one thing we don't tend to talk much about is you're working in negative ion, and I know the instrument mass specs. I hope they've improved, but it used to be that negative ion was very temperamental from instrument to instrument, even on the same platform. Do we still have that as an issue, or is that something that's largely been resolved by the manufacturers?
We don't have that as an issue, because we isolate instruments when it comes to the oligo work in negative mode. So if we're using such and such an ion pairing reagent, we can mix projects on that instrument as long as we're using the same mobile phases, and we don't really see any issues. But yes, if we were to hop back and forth with different projects, where we're conditioning the instrument again, and we're exposing the instrument to different extractions and different mobile phase modifiers, yes, then it can interfere with negative mode.
And Dominic, just for your information, this is one of those instances where the change in drug design really impacts us as bioanalytical people. We talk about small molecule. We routinely would switch one assay in for another without really doing anything special. It's just your one assay finishes, you then move on to the next. There might be some change of solvents and stuff, but it's not huge. But oligonucleotides are really, they don't play nicely with other assays at all. And so that's why we're talking about these isolated instruments that you can focus on the oligonucleotide assay, because switching from oligos to something else means a whole lot of downtime between assays, and for a CRO, obviously, that's not where you want to be. So that's a challenge for the operations team, is how do you accommodate the need for oligonucleotide analysis, but also ensure that the instruments are used efficiently, knowing that oligos are a tough kind of assay to slot around everything else.
Yeah, John, I don't know if that was a question in there for Bryan, but I'll just say I was really enjoying listening to you two talk, because I feel like I'm taking us back a little, so that's why I'm going to be a little more quiet. But I know our audience is definitely, me personally, I'm anxious to hear about ADCs. But I think your question to Bryan was, how do you manage oligos, right? Like, they're tricky. They're going to throw up your instrumentation. Is there a way? Is there a secret sauce behind it, or is it just, that's why we got a fleet of instruments, right, John?
It might take us because we have a fleet of instruments, but I'll let Bryan chime in on that.
Yeah, it's, as we were talking about, it is very challenging, but we've gotten better and better in our approaches to it, and isolating instruments with specific ion pairing mobile phases, trying to use the same analytical column when we can, that's already conditioned with the ion pairing reagent, we were able to put more projects on one machine and have less downtime, and have more control over the assays. That's what it's all about, is having control and making sure that um, we have verification solutions of the drug that we check before we put on our assay. So if we have if we're putting on another project on that machine, we'll do an injection of our drug and meat solution to make sure that we have the right response before we actually put the run on. So it's all those little checks and balances that we've learned over time that avoid downtime and lead to our
Yeah,
success.
Brian, there's an oligo. Are you aware of the? We're on a lot of APS committees, and I can't keep track of it. I know at one point we were on the oligo. Are you on the oligo at all? Or are you aware of this APS focus group on oligos?
I am not.
Okay, so Brian, I'm going to volunteer you to go and give a talk at that group and get you on that group. You okay with that? Because what you just said, I don't want. We don't have to rehash it, but I think what you just said about how to help maintain downtime on oligos, or how to maintain your instrument's functionality, how to prep it and dedicate it, is something the world might need. Unless you tell me, not down, there's already stuff out there, but in a regulated environment. That's the key, Brian, right? That's what you're talking about, how to do it in a regulated environment. And trust me, we might think, who would want to know this? But Brian, the world definitely wants to know that, unless maybe we research it a little more. I'll take that offline with you about it, but I think it's a great thing to maybe share as a poster, or even just, I got to imagine there's some group out there talking about oligos, and we should be more actively involved. John, I'm going to pass it over to you, but Brian, great discussion there. Really cool how you've developed what I'll call a process to help me ensure that you can provide good, reliable uh, uh, oligo data, and make it effective and cost efficient for the client. That's important. Meaning you can't just keep throwing instruments at this thing. It's just not sustainable in terms of the sheer cost of them, and then where you got to put them. So, maximizing it, how to be efficient, how to do all that. Not so simple. I don't think it's a simple thing, and it's definitely something that we're fine sharing that type of stuff, Brian, because good luck to everybody with trying to figure out the methods. But maybe I'll pass it back to you.
Yeah, I think that's good. I think let's move to the ADC piece, because obviously ADCs have become a big part of what we do as a company, and we've talked largely about the large molecule piece, whether we do it by ligand binding assays or we do it by hybrid LCMS. And the payload is fundamental to the success of an ADC, but we sort of almost take that assay and the method for that small molecule payload for granted. But what we've been finding, and that's a lot of the reason why I wanted to have Bryan join us on this podcast, is that actually it's much more complex. It's a much more complex issue, and so there is a lot more needs to go into development and validation for payloads. And I'll let you talk as much as you want, Bryan. I'm not going to steal your thunder on that.
Thank you, John. Yes, so we've been doing these assays for years now, and what we've learned is it's not just about developing the payload assay, as you were mentioning, John. It's also taking a look at the ADC while we're developing that payload assay. So what we found is that we need to take a look at the blood collection. We need to take a look at the plasma if we're doing a plasma study. We also need to look at the analysis, what's going on in the extracted analytical plate. And then we also need to assure that we're covering ISR, and there's not any challenges that are going to occur in the ISR, or be ready to have that toolkit if we need to troubleshoot ISR. So in terms of the blood collection, what we found is it's important to characterize the ADC and the payload separately. So our typical approach is that we'll take the ADC at Cmax and we'll look at it in the whole blood stability assessment. In parallel, we'll look at the payload by itself, the same time points, the same temperatures. And what we have found in some of these projects is that one of the either the ADC or the payload will have a shorter collection time or a different temperature that needs to be obeyed, which, if you can imagine, in a preclinical study, say a mouse study where the sample volumes are short, right? We're in a position sometimes where we have to share samples with another lab, and because at KCAS Bio we do everything under one roof often in LBA or in hybrid and in a small molecule LC-MS lab, where we have to share the same sample. So that whole blood collection has to be good for both of us. And, that's something that's not being looked at, especially it's not being looked at, I know, especially is if the ADC is being assayed at another lab. And so at our lab, we're talking to each other and we're talking to the other department. We're asking, what's your stability look like? What are you seeing? And as I said, in our assay development, we're taking that ADC, we're running it parallel to our free payload. We're characterizing the blood collection and making sure that the blood collection does not have to be different for one or the other. And sometimes it does, and so sometimes one of them will dictate what the other collection has to be. The other important thing we found is taking a look at plasma stability. So once the plasma is collected from the whole blood, we're looking at both, again, the ADC and the free payload, and we're doing a time zero. And the time zero is important because for the ADC, we want to see, is there any release at time zero? And if there is, now we carry on to benchtop and freeze thaw testing to cover the sample handling in the lab. And we're doing that in parallel. We want to make sure that, and we have seen this, that sometimes the ADC will actually start releasing the payload at a certain duration on the benchtop at a certain temperature, and also during a certain freeze thaw cycle where it impacts the free payload assay.
If I could just step in, Bryan, quickly, that's an important thing to focus on because the concentration of the free payload is very, very low, whereas you got a high concentration of the ADC, such that if there's any breakdown of the ADC, it can drastically affect your payload assays, and again, that impacts the reliability of the assay that you're developing and validating.
Yes, we found it to be very important. And if I back up to the blood collection, also we've found in some projects that it's concentration dependent. So if you put in a lower amount of the ADC or a higher amount of the ADC, then you have a different outcome. So it's important that we do our homework. We're talking to the other lab, making sure everything's straightened out ahead of time so we don't have trouble down the road. The other item we look at is the assay plate. So if you can imagine, if you have extracted some samples for the free payload, but you have some existing ADC that's also been extracted in your plate, even though we're not looking at the ADC, it can release some of the free payload over time in solvent in your assay plate. So we take a look at that. And we also characterize the ADC in solution. So we'll take a stock, we'll look at it, and we have found different levels of impurities with these ADCs. And sometimes that becomes a consideration we have to make when we're designing our assay. It's important so we know what we're dealing with ahead of time as we approach these assessments, and we have an idea how we can mimic the study sample the best that we can so that we can have the best outcome when we are approaching the ISR. So all this work, is supporting mostly like the blood collection in the ISR. We want to have reproducibility when we grab those samples and we have to analyze them again. We're obeying the time that we're handling the samples, the temperature that we're handling the samples, and how many times we're thawing them out.
And it's a great again, similar to the oligonucleotide. It's a great illustration about how these more complex therapeutic entities are actually making our lives as bioanalysts much more difficult, because like I say, we know the payload's that small piece, but ultimately you can't think of it as a small piece. You have to be thinking about it as the bigger entity, and so you're doing a much more thorough investigation of the assay to make sure that it's fit for purpose downstream.
Yes, exactly. With that blood collection also, if you imagine you're doing a PK study and you might have 120 minutes stability from the free payload, and then you find out by taking a look at the ADC that by 15 minutes and 30 minutes and 60 minutes you have different amounts of release. Well, now if the lab is letting some of the in vivo techs are taking samples at 30 minutes, or someone comes along and whatever, has multitask and collects at 120 minutes. No, that could impact your PK results too. You're getting some variability in your time points. It could be okay on our side, but for the client, it's not a tight data package if you're not understanding and doing all the homework.
Great, Bryan. I mean, you're some of what you just described is what separates us from our competition in a big way. And it's always refreshing that I sit out there and always try to articulate it, but you nailed it, Bryan. You got to look at these ADCs in a way where what if something does go wrong? How can I think of it as engineering or designing the sample processing to avoid having to draw three different, tube types. And then even just in, some of the chemistry has changed in this space, but there always is that need to maybe have to literally, and we can do this under our roof, just pass it from the small, you guys kind of have to do that upfront small molecule piece, right? That is, not that there's not instability elsewhere. And then biotransformation, Bryan, we didn't even talk about that. That's all the rage these days. I just think of that as biochemistry. But all of that requires a lot of just years of, I think, CRO experience in both LBA and small molecule to achieve that successfully. And then you said something incredibly powerful about, like, I think of it as, if we have the ability to do lead candidate selection in the discovery space for your ADC, man, that helps us, right, Bryan? It helps a lot with knowing if this team had seen this in the total ADC assay by LBA. And that's what you were kind of articulating before, right? So that there is the ability for somebody to come to you and say, yeah, Bryan, I do see this and that. And that helps. That helps a lot, right? If they know there's perturbation in the total antibody measurement versus the total ADC measurement, putatively because of some biotransformation or some falling off or whatever it might be. If you don't have to do, if you go in knowing that, it does help, right? So
Yeah.
all of that is what why we're I don't know. There's no measurement here, but I believe you'd be hard pressed to find a CRO in the world that can support ADCs the way we do and have supported just the sheer number that we have. I think we're getting up there on validation of protein LCMS methods for supportive ADCs like no one in the world. And why I say that is because EBF showed 27 case studies for protein mass spec, and then I showed 5. So that's incredible. That was like a consortium of, I don't know, 8 or 10 companies that had 26 case studies or 27, but I just showed 5 of ours. I was pretty proud of that, and more to come. We'll share more with the EBF, but I'll throw it back to you two.
And
And
I'm gonna
this has
add
been great.
one more thing. I think because the development process for the payload is because it's become more extensive, it means that what we are more involved in the manual preparation, and so it yes, the preparation for samples and support for clinics and support clinical trials can be quite complex, but we know that they're likely to push back on some of those parameters because obviously it's not just a routine take a sample and store it like a normal small molecule drug, because there's multiple steps. It has to be on ice, has to be flash frozen with dry ice, whatever any of these parameters are. If the clinic comes back and says, are these necessary, we have the data to back up and say, yeah, you absolutely have to follow this to make sure that the process is followed, so that the sample integrity is good, so the data is good, to help then, like Bryan mentioned, to ensure that your PK data is as good as it can be.
Yeah, it's that blood stability. We get asked all the time, how should I store my sample? I'm like, well, give me a drug and we'll take a look at it, right? Like, we can't read that.
Yeah.
But Bryan, maybe you want to add something here, but it's been great. It's been really good, Bryan.
I just want to emphasize that it has been a great outcome with our clients to have everything under one roof, because I often find myself in these ADC projects, like I said, talking to the other lab in our company, talking to LBA or talking to hybrid, and seeing what they're experiencing, and then looking at the protocol and making sure that these collections make sense, and making sure everybody's on the right page.
Yeah, I think that's important because we're all talking, then we are treating we're really thinking about how best can we support the entire entity and the assays that are needed to measure that drug, so the payload isn't just a forgotten appendage. It really is integral to how we think about these drugs and how we approach the
Well, it's one of our pillars, John, right? So that's the collaboration, right? So we talk about K is knowledge, C is collaboration, A is accountability, and S is service. And Bryan, you just articulated collaboration at its finest. Bryan has an idea, John, maybe you guys have ideas of the small molecule you synergize. You go over to Dawn and Ratna and you start synergizing about hybrid. Then you come over and talk to Lindsay, Sheck, and myself, and we synergize on the LBA. That's collaboration. Do we do it great and it's all worked out? Well, yeah, we do it very, very well, but that's still growing in our organization. We're getting better every day with what I just said, right, guys? That is a big part of why people should, hey, bring your ADC to us. We can help you with it. It's a really, and that's been organically growing for 5-6 years. Like, Bryan and John, you guys are over here learning more and more about ADC small molecules, and you're not living in a vacuum to get there. The LBA team, same thing. Hybrid, the protein mass spec team, all three of us are every day going, oh man, maybe we're learning more about this at an LBA perspective. Hey, now we're starting to think, I wonder if the small molecule team could use this information. We're proactively starting to collaborate that way. And that's palpable. That's part of our job, by the way, John. That's more to come in 2025. But Bryan, I hope that I can hear it in your voice. Like, I wouldn't have been able to solve some of my problems if I didn't have teammates coming in going, oh yeah, we already looked at that, right, Bryan? It just helps you reduce, because at some point you're looking at, man, I got to throw the kitchen sink at this thing, right?
Exactly.
So I think we obviously this has been a great discussion. Thanks very much, Bryan. Really appreciate your time. Is there anything more you'd like to add, and then we'll move to close out?
No, I think that covers it. Thank you.
No, I love this. It's a great topic. ADCs we're going to keep talking about more and more. There's no doubt about it. So, do you want to take the lead on the next piece, Dom?
Yeah, this is the hot seat, Bryan. There's not anybody in the hot seat, but the first question, and it's John and I, it's near and dear the most important question. What is your favorite podcast? No, I'm just, but what is your favorite podcast, Brian? You got one for us?
I enjoyed podcast 73, the parallel evolution of industry in KCAS. Like I said, I've been part of KCAS Bio for 5 years, so I've seen all this growing right in front of me and been a big part of it. So I'm really proud of that.
It's awesome. That's awesome. And we enjoyed doing that one, and more to come. We're going to continue to have more series around that. But I won't have to grab the spotlight here, John. I don't actually, but for some reason I don't see the hot seat questions. What's up?
It's right at the bottom of the agenda. So the next one is favorite dish to make.
I could say I've mastered lemon chicken. So I do lemon chicken with real lemons, and I do a certain kind of reduction sauce that I'm proud of, and that's my favorite.
Man, that sounds delicious. You got capers on there or something? No capers.
What is that?
You use capers at finish it with a little capers or anything like that?
I don't do like the piccata angle on it. No, I don't use the capers, but I do I use fresh parsley from home.
Okay.
And whatever I can add from home.
Little breaded, right? That is like definitely to me one of the most challenging dishes anyone's mentioned. That sounds really hard to perfect, right? I'm pretty I haven't even, as an Italian, Brian, I haven't even tried that deep a dish, but I'm getting there. I think I'm getting up the courage. I'm starting to do some reduction myself, but that I love it, and I might hitch up for a recipe. But
it sounds delicious, I have to say. Yeah.
I'll go with the next one, and that was really cool. I imagine you can get fresh lemons all the time and stuff like that down in Florida. So, tell us about your favorite adult beverage.
I won't refuse a Cabernet from California. That's my favorite.
Just don't go sideways on us, if you know my reference, right, Brian?
Yes.
No, I don't. I truly enjoy just about all liquors, as anybody who listens to the podcast knows. But I think you were very specific with a cab from California, so you probably got some up there that you know. But wine is so vast. I look at it this way. I don't know vineyards and things like that, but in grad school, the guy I was the principal investigator, he used some of his biotech funding money to do a party every year, and he had a wine tasting. So for 5 years, I learned how to wine taste. So I know a little bit about things, but the long and the short of it is I couldn't tell you much other than this isn't good and this is good. But isn't that how everybody drinks wine?
It's really quite important.
Yeah. And then I gauge by how bad my head hurts the next day. That's always a big one too. Yes. Like I'm not getting that, Brad. But I don't know, John. I don't know if you have any comments on the wine thing, but we can.
I think Cabernet and probably Zinfandel are my wine grapes of choice. But yeah, I'm perfectly happy to have a glass of wine at any time.
I just love French table wine. That's my favorite. I really do enjoy that. So
I'll be honest. After a trip to Scotland, I now have more single malts than I know what to do with. So we might divert into that over Christmas, and sort of going in that direction.
I got some single malt with some pineapple in it. It's okay actually. I don't know what made me buy this thing, but it's decent. And you got me thinking about that. I'm not one to ever want to see any sort of fruit in my. Liquor.
I'm not convinced by that.
No, like those apple ones. Like if you ever have a Crown Royal apple, those are gross. But Brian, we're going to turn our attention back to you.
Just very quickly on the single malts. So obviously, I was in Edinburgh with my wife recently, and we went to one restaurant for lunch, and I was on antibiotics. I couldn't drink anything, which was a bit of a pain. But it was like pretty much at the end of it, most tables. Someone ordered a whiskey because obviously it's Scotland, so it's you do that. But the restaurant did, they would bring out the bottle, pour you a glass, and then leave the bottle there. So it was really interesting to see all the different whiskies that people had ordered and try and identify who was drinking what from the menu. So actually, one of them that we saw there, we then tried. It's one called Bunnahabhain 12-year, was fantastic. So we picked up a good tip from that. Anyway, not to detract, I'll stop talking. What's your weekend plans as we start to move towards winding down the year?
Oh, thanks for asking. So this time of year, we're going to go check out the Christmas lights in the center of town, and we have shopping to do.
I think that is well said, Brian, and I think all of us fall into that bucket. There is some light. I have my couple neighborhoods my wife and I like to drive through. It's not necessarily the center of town, but a couple little cul-de-sacs. It looks like winter wonderland through there, and there's a couple other things you can drive through and pay. You got to reserve and go see these lights in like Longview Lake, I think, in Lee Summit's where there's one. And then, of course, the Christmas shopping's big on the list this type of year. You bet. This time of year.
We done the shopping. We got the tree in. The tree is now sitting in our front room, but with nothing on it. Well, the cats get used to it, and then next couple of days we'll probably start decorating the tree. So, but the cats have calmed down around it.
Yeah, my tree went up a couple of weeks ago, holding up pretty well, and the wife's got the presents around the tree. And my son comes home from college today, so he'll be home for the month, and that's always exciting. But love Christmas. I go to Nebraska every year. I love it. It's a great decompression for me. I get to go and nobody knows who I am. I don't have to do much. I catch up on TV. I exercise. It's fantastic. I can't ask for and then get to spend good time with my wife's family, like Christmas Eve. 40 or 50 people. We rent out the same hall. Been doing that for a long time. So that's a fun night. But I don't know if you guys want to share. Otherwise, we can wrap up.
I think we should wrap up and move on. But thanks very much, Brian. I really appreciate you joining us.
Yeah, like I said, thanks for having me.